Okay, so this is our video on how to complete the cell cycle checkpoints in Cantor if you were not in class when we did this. So you will need to write a description, it says here at the bottom, for each checkpoint on the cell cycle on the slide of what might happen if that checkpoint were missed, okay? And so you're going to read, I'm going to show you how you can actually read, okay? Checkpoint or miss, not done during the normal cell cycle. Please take a look at what the checkpoint is looking for at each of the 3 areas and discuss what might happen to a cell if that checkpoint were bypassed and the cell continued on without confirmation that those points were okay. If you need more space in your text box, please make your font smaller. Once you have finished all 3 explanations, please drag and drop them onto the slide as close to their checkpoint arrows as possible. Okay, so you have 3 boxes here, okay, and to figure out which one it would be, so this would be the metaphase checkpoint, you're going to click on this link up here. This is a whole reading of checkpoints and how they're missed. So you will click on here. It'll bring up your little kind of preview thing. You will click on Open Preview. After you finish clicking on Open Preview, what you're going to click on now is right here. It says Open Document. It's not open in New Tab. Okay, and then you're going to click that open, and it should open up this whole reading for you. Okay, and you can go through them. So it talks about the different checkpoints here. And it says G1, G2, and spindle checkpoint, which you have 3. So you're going to go ahead, you're going to read through, okay? And it also shows you where they should be, okay? So if you look, you've got a metaphase checkpoint, G1 and G2. So you have those on the reading as well. The spindle checkpoint is the metaphase checkpoint, okay? Just a different word for it. Okay, but you're going to read through each of these checkpoints and you're going to decide what happens if it was missed. So what is it that's going to happen if these things are not checked? What could happen in that cell cycle? Okay, so it'll read about G1 and then you have G2 here and then you have your spindle or the metaphase checkpoint, okay? So you can see here, so let's look at this metaphase real quick. I'll do this as an example for you, okay? And it says the metaphase checkpoint is also known as the spindle checkpoint. Here the cell examines whether all the sister chromatids are correctly attached to the spindle microtubules. Because the separation of the sister chromatids during anaphase is an irreversible step. So remember those spindle fibers attach and it pulls those sisters separate so that they can go to the different cells. If it doesn't, you can think about, what would happen? The cycle will not proceed until all the chromosomes are firmly attached to at least 2 spindle fibers from opposite poles of the cell. How does this checkpoint work? It seems that the cells don't actually scan the metaphase plate to confirm that all the chromosomes are there. Instead, they look for straggler ones that aren't attached, that are in the wrong place, okay? If a chromosome is misplaced, the cell will pause mitosis, allow time for the spindle to capture it for the stray chromosome, okay? And so then you go back, what do you think would happen? So if it didn't check to make sure everything was attached, if it just had a stray, what are some things that might happen? So you can think, well, if they don't separate, then you're going to get 2 copies, right? You're going to get 2 copies of those chromosomes over there. And we talked about you don't want 2 copies because then you can have genetic issues, right? So some things that could happen, and you can, I don't want to underline, so I'm going to get rid of the underline. You could, okay, have 2 copies of the same chromosome in one cell. This can cause genetic abnormalities. Okay, if I can spell, I'm going to bring it a little closer so you guys can see my typing as well. So you could have problems within that cell for genetic abnormalities, right? You can also, here, let me go up here. You could also have, we'll scoot over, okay, also could cause a problem in division, right? Which can lead to fast division or cancer, right? For example, tumors. So what can happen is you can get a problem with division. So then what's happening is there's too much stuff in there. It's causing a lot of problems, okay? You can also have, let's say, if you have too many chromosomes in there, okay, you could have your body start attacking it. And so your body could attack the cells and kill them. So if you need them, then you may not get repair, okay, right? Which would interfere with repair, or maybe or for some other animals, regeneration, okay? All right, so I'm going to get rid of this so that I have a nice thing. Then you're going to drag it over. So I gave you a little bit of enough room. So the boxes are the same. So I'm going to go ahead. I need it to be a little bit smaller. So I'm going to go to 11, which is then because I resized my box and I shouldn't have done that. And then I'm going to resize my box. Now that I've made this one, I'm going to take this and I'm going to drag it over the metaphase checkpoint area. Okay. Okay. And so then you can see that it is there. Okay. You can actually, I'm going to put it to the side just because then I can see it and read it. Okay. All right. So that is your metaphase. So that one's done for you. Now what I want you to do is go back and I want you to read about G1 and G2 checkpoint. And then when you are done, you are going to link this, as it says here, into your notebook. You're going to link it on